Good morning, everyone. Thank you very much to Joanne and the hospital for inviting me to speak here and to be part of such an amazing faculty. I feel like the fake person in the faculty with not so many accolades as the others. But I want to talk to you today about overactive bladder and chronic infection. I run a unit which is a tertiary referral centre for chronic UTI. I have done a PhD with James Malone Lee. Some of you may know him. And I think it's very important that we don't forget and think about infection. Some of the data that I'm going to present today has helped us. It's prospective data and some retrospective data has really been very important to inform us with regards to an RCT that we are now planning. Okay, so now we all know the definition of overactive bladder, and we love definitions in our line of work, maybe a little more so than other specialties, but we know that overactive bladder is all to do with urgency. There's usually frequency, nocturia, and it's with or without incontinence, as Joanne mentioned. But of course, life isn't so categorical. And if you think about it, there are so many conditions which will have very similar symptoms. You know, we have patients with bladder pain who will have these symptoms, interstitial cystitis, UTI as well. So we've got to be mindful of that, you know, these things are not so straightforward. And sometimes it's very difficult to put a patient in a category. But of course, when we make the diagnosis of overactive bladder, we try to exclude infection. So the question is, if someone we think has overactive bladder, how do we exclude infection in that patient? And that was largely the basis of my PhD. So, in the UK at least, the way that we exclude infection, if we start from the bottom, if you go and see your GP, you will no doubt have a dipstick test done to look for infection. The GP may then send your urine off to a laboratory, and the laboratory usually will screen the urine. So they'll stick it under a microscope and they'll have a look for white cells or pyuria. And we use a threshold of about 10 white cells per microliter to say this specimen is screen positive and we should try and submit it for a urine culture. The criteria we use in the UK for positive urine culture was described by Cass in 1957. And he actually studied women with pyelonephritis and normal controls. And he concluded that more than 10 to the power of 5 colony-forming units per mil of a known urinary pathogen means that you have an infection in that group of patients. So these are the sort of current criteria that we are using. Now, as I said, Cass described this in women with pyelonephritis. And again, we're coming back to categories because we love categories. So we have to be mindful that when you do a test, of course, it depends upon the population and the sensitivity of that test in that population. So Cass described women with pyelonephritis, and he said that 10 to the 5 is likely to indicate an infection. Then Stam later on looked at women with acute UTI, so with dysuria frequency, who were attending A&E. And he concluded that actually Cass was probably missing a proportion of women, and he suggested that 10 to the 2 colony-forming units per mil is probably a better threshold for acute UTI. And this we call the spectrum effect. But what about women or men who have chronic symptoms? Have we looked at that population? What do we know about them? And can we still categorize them in the same way? Now, the dipstick has been the most popular marker or surrogate marker of infection that we know. And of course, it's been studied on the basis of the MSU culture, so the 10 to the 5 colony-forming units per mil. If we look at pyuria, pyuria was studied by someone called Dukes in 1928, and he actually looked at the excretion of white cells in the urine in asymptomatic men and women. And he looked at the mean excretion rates, and it's probably quite an overinflation, because if you look at this curve, you know, it's got quite a sort of a variety of points on there, but he said that the normal excretion rate of white cells, so in, you know, asymptomatic men and women, in men it's about 1.6 white cells, and in women 5.4. It's probably an overestimation because he used the mean. Probably better to have used the median. So it's likely that the excretion rate is even less than that in normal, shall we say, individuals. So he actually used 10 white cells as a criteria. Again, love thresholds. So we make a cutoff, 10 white cells, easy number to remember, that if you have more than 10 white cells, it does actually correlate. You're more likely to have a positive MSU, but it doesn't correlate the other way around. So if you have a negative MSU, it doesn't mean that you have less than 10 white cells, if you see what I mean. So again, we come to categories. And what do we know about other groups of patients? Are men the same? Are women the same? Are young people the same? Are old people the same? Are chronic patients the same? We don't know. So I started my PhD with Professor Malone Lee, and we were a referral centre, tertiary referral centre for patients with overactive bladder. And we made some observations about these patients. So there's a whole group of these patients that will not respond to anticholinergics, and we know that you don't get tachyphylaxis to these agents. You know, you don't form a resistance to them. So as long as the patients were compliant, why were these things not working? And also to Botox. And we thought about infection, and we thought about, you know, well, we're trying to exclude this in these patients. Are we missing something? So along with culture and dipstick, we started to microscope the urine of these patients fresh. And about a third of them had pyuria, but 2/3 of those had a negative urine culture. And that was actually the group of patients that we started to study. Now, very exciting for me, one of the things that I did in my PhD was follow urine around the hospital. Very exciting. And we published a paper about the standard test that we use to exclude infection in these patients. And we used our gold standard, which is supposed to be MSU, to donate an infection. And we used the 10 to the 5 cutoff. And what we realized was that the dipstick test is actually very insensitive when we look at infection. And although not perfect, if we microscope urine fresh and we look at white cells, that actually it's a better marker of infection. Now, I say that I followed urine around, and this is an important point because I don't know about other countries, but in our National Health Service, you'll take a urine specimen from a patient. It will take between 2 hours to 5 days for a specimen to reach a lab. And I sat these specimens on a bench and looked at them over time, half an hour, 1 hour, 2 hours. And it's very interesting because the white cell signal deteriorates very quickly. So you have about 2 hours to look at a specimen to really understand what is the white cell count in that specimen, and after that the white cells implode. So it's very, very important that we understand the deficiencies in the pathway of doing an MSU culture. You can imagine 5 days later, what can you get out of an MSU culture? Even if someone plates it, I don't know. So we just have to be very mindful, and patients say this to me all the time. My dipstick was negative, So I was told, you don't have a urine infection, or I will constantly get culture results which will say the screen was negative, so an MSU culture was not submitted. And of course I know how long it took for that urine specimen to get to the lab, and that any white cells were likely to be depleted. Okay, now, when I was looking at all of these urines under the microscope, I submitted them to gram stain. This is a gram stain from one of our patients. And again, science is about observation. And we made an interesting observation, which was that in the urine, the bacteria love to stick to the cells. So when you find all these bacteria clusters, you can see that that's two epithelial cells, and the bacteria are stuck onto the cell. I guess that makes sense. That's how you would cling on to a high pressure system which is always being flushed. So we proposed, well, when you submit a urine to culture, you're looking at the urine, but what if we separated the sediment out? Would we get a higher harvest of bacteria? And that's what we did. We spun out the urine with a centrifuge, got rid of the supernatant, and plated the sediment. And yes, lo and behold, you get a higher yield of bacteria. So, what does this mean about our standard routine methods that we use to exclude infection? And again, we have to be aware of the limitations of our tests. We love to believe in tests, but we need to make sure we understand how it's done and what is its limitation. Now, another interesting thing. Well, I think it's interesting. So, in the NHS, there are a number of ways that you can perform an MSU culture. And that's pretty standard. So there's a whole document produced for laboratories that you can do your culture in X, Y, Z, or AB way. And it's supposed to yield the same result. And it's very technical. It's to do with what size of loop you use to plate the urine, and this sort of thing. So we tried all the different ways, of course, on the same urine specimen. And again, interesting, depending on what size loop you use, depending on if you use the sediment of the urine, you will get a different result in the same urine. So if a patient goes to hospital A and then goes to hospital B, it's possible that they will get a different result depending on the technique that is used to culture the urine, and even the medium that they use to culture the urine. So in the NHS at least, the culture medium that they use is very specific to certain pathogens. So in that process, we exclude everything else. We're very, very focused on E. coli and picking up E. coli. Okay, now, this is a diagram cartoon from Holtgren et al. From Washington, and they looked at UTI, and they used a mouse model. So they infected these poor little mice with E. coli, and then they sacrificed the mice at different times, and they looked at their bladders to see what the bacteria were doing. And it was actually quite remarkable. So for a long time we believed that bacteria were swimming around in the urine or sticking to the uppermost surface of the urothelium, and that was about it, and that actually we flushed quite a lot of this stuff out. But what Holtgren found was that the E. coli were actually invading the urothelium. So if you look at the diagram, it sort of goes from the top. So they were attaching. Oh, I could use this. So they were attaching to the urothelium, and they were actually invading all the different layers and going deep inside. And he described something he called a biopod, which was a community of bacteria, which was then developing. And it was sort of walled off. So these sort of deep bacteria that are going inside, they were evading immune detection. And for the mice. And so these mice were resistant to treatment with antibiotics. And then what was happening was that this biopod was sort of bursting, and then the bacteria were just sort of swimming back and setting off that cycle again of invasion. Now, of course, mice are very different from human beings. So we thought, is this happening in our overactive bladder patients? Are we missing infection? Because actually, these bacteria are very clever. They are invading the urothelium and avoiding detection, and it may not be so easy to culture them. So we looked at our patients. Now this is an image from one of my colleagues in our lab, and he's used confocal microscopy. And this is from one of our patient samples from their urothelium. And you'll have to take my word for it, but along with the confocal microscopy and some stains, he has shown that essentially these bacteria are actually within the cell. So in microbiology, it's very difficult to show, is something just stuck to a cell, or is it inside, is it actually inside the cell? And for the first time, this confocal microscopy has allowed us to see, so this, essentially the green bits mean that the bacteria are within the cell membrane. So even in human beings, and this is an enterococcus, so in human beings we are seeing that these bacteria are invading the bladder and sticking there. So again, difficult to treat, difficult to detect. So what does this mean? Again, we just have to be alert to that really we must make sure we're not missing infection. Okay, now, so we looked at normal volunteers or asymptomatic volunteers. We looked at their urine, we cultured their urine and submitted their urine to sort of more complex techniques of detecting bacteria, looking at DNA, bacterial DNA. And it's very interesting. So if you look at those two pie diagrams, you've got OAB patients on one side and you've got control samples on the other side. And what's interesting is that everybody has bacteria. Everybody's got some sort of bacteria in their urine. And yes, there are differences. There's more of some and less of others in the LUTS patients and the control group. But by and large, everybody's got bacteria. So what does that mean? Certainly when I was at medical school, we were taught that the bladder is sterile. And I think more and more evidence has now been produced about the microbiome in the bladder and that it is not sterile. So a little bit like your mouth, really, that there's lots of bacteria living in there. But what is the difference between them living in harmony with us one day and then the next day causing us a problem? We don't know. This is a similar representation of the same thing, really, in that, yes, there are some slight differences, but essentially the control group and the patient group all have bacteria. And like I said, you don't need to take my word for it only. There's a huge interest now in the microbiome and how it impacts how we are treating patients with bladder symptoms and bladder problems. Okay, so what have I told you so far? So overactive bladder might be secondary to a UTI that we haven't actually picked up because we can see some of the shortcomings in the tests that we use to detect infection. Now, we talked about pyuria of more than 10 being significant, but like we said, this was a cutoff. And actually, it's more than likely that the actual excretion rate of white cells is much less than 10 in a normal individual or asymptomatic individual. So we don't actually know, even if you have between 0 to 9 white cells or 1 to 9 white cells, really what is the significance of that? And certainly in our practice, we can see that it can be quite significant in terms of symptoms. We know now that the microbiome is very complex. I show you those pie diagrams and those graphs, and for those of you who are awake, you probably saw that E. coli is not the predominant bacteria that is grown in these patients. Remember, these patients are chronic patients, so they're slightly different from the acute patients that you might see in A&E. And we were surprised that E. coli is, not the favored pathogen. And that we can now understand through animal models and actually through human models that bacteria may be resisting detection. They may be forming these biopods or biofilms and intracellular reservoirs inside the bladder. Okay. So how are we going to treat these patients? The answer is with great difficulty. Now, I've got this little cartoon drawing again, and it's just one thing I want to point out about it, is that if you imagine that's the urine and this patient is infected and there are some white cells produced here, and you've got the sort of top layer of the urothelium which faces the urine, and it's got all the sort of protein coat on it, and you've got all the different cells layers, and you've got the baby cells here. There's one other interesting phenomenon, and I'll use this as little green blob as an example. In microbiology, there are a bacteria which we call persisters, and essentially they will invade any sort of epithelium, and then they will become quiescent, so they stop dividing. And we don't know what is the mechanism of this, but because they stop dividing, antibiotics can't kill them, and they evade detection. So you may have a patient who has a UTI, goes off to the GP, has a course of whatever, antibiotic, and then we'll go back to 2 weeks later because the persister for some reason has woken up and decided that is now going to start dividing. So persisters are also a problem. Now, this urothelium in the natural life cycle, you can imagine our bodies are very clever. If we have something invading, what does your body do? It will start to shed this epithelium or urothelium. And we see that in the urine. Now, initially, when I started to look at urine, we used to think that the cells that we found in the urine were contaminants. And again, this is what we were told. These are contaminants. They come from the vagina, and they are vaginal squames. So again, we put this to the test, and we stained those cells with uroplakin, which is very particular to urothelium. And actually, we were very surprised that 80% of the cells that you find in a urine specimen on microscopy are actually urothelium. So it's very interesting that, again, patients shed a lot of urothelium. So you'll find a lot of epithelial cells in their urine compared to the volunteers. So it takes quite a long time for you to shed all of this epithelium. So you can now build a picture of the problem. Difficulty in detection, persisters, the fact that your urothelial layers will take probably several weeks and months for you to shed them, and during that time the bacteria are likely to just swim around and reinvade the urothelium. So you can see how chronicity and recurrence can occur with infection. What predisposes some people to have a chronic infection? Don't know. Why do some women have a UTI and drink some water, a little bit of extra water, and then they're okay? And why do some women have this persistent problem? Don't know. So, I like this slide because it's just so telling about how patients view us, really. By the time I see a patient in our chronic UTI service, they've been around the houses for about 6 years. So they've been to various doctors, had various tests, and had various treatments. And really, the challenge is that, one, they don't trust me and think I'm going to fob them off again or do something terrible to them. But how do you include yourself in that circle of trust again? And I think the answer is to have in your mind that tests are not what they're cut out to be, that we do have to take it seriously. We have to consider this at least. That's the first step. Consider that this is a potential diagnosis. Sadly, understand the limitations of the MSU. and actually trust the patient. And I say that because we published a paper about symptoms and how they relate to chronic infection. And we know a lot about acute UTI. There's a lot of patients who will, it's fairly straightforward. They will have frequency, they will have dysuria. But in chronic patients, and again, we don't understand why, but the symptom profile is slightly different. And what really stands out is voiding symptoms. And I have to say, we don't classically pin down women and ask them about hesitancy, slow stream, intermittent stream. This tends to be the remit of the urologist looking at prostates. But actually, voiding symptoms are very, very important in this group of patients. Okay, so I asked the question, how do we treat these patients? So we do have some data from randomized control trials, but really the data is sparse. There are a couple of studies about hyaluronic acid, I can't say it, so various bladder installations. There are some studies looking at antibiotic efficacy. So if you treat patients who have negative cultures or indeed positive cultures, what are the outcomes going to be? Now, this is an interesting study from Stam, and he treated patients who were culture negative but who had frequency dysuria and pyuria, but they particularly had urethral pain, and he treated them with doxycycline for 10 days. And he got good results in terms of treatment resolution. I think 75% were resolved. However, there's no long-term data to support, what is the long-term outcome for these patients. Now, sadly, if you treat patients with antibiotics, there's a failure rate. So if we, in the NHS, we treat patients for 3 days to 7 days. 3 days is very popular. But we know that there will be a symptomatic and a microbiological failure. When I say microbiological failure, of course, I mean cultures. And that will be within 2 weeks of having a 3-day treatment or even a 5 to 14-day treatment. And that persists. So if we treat patients between 4 days and 4 to 14 weeks of treatment, again, there is an inherent failure rate. So when you stop treating these patients, essentially their symptoms return. What we do know, though, now is, I've told you that MSU can be unreliable, and microscopic pyuria is now standing out as probably the best way to detect infection in these patients. But of course, it needs to be done fresh. Like I said to you, it will deteriorate within 2 hours. And that's not accessible to everybody. I know it sounds very straightforward. It's just a microscope. Look at, you don't have to have any special instruction, really. And microscope is pretty cheap, but lots of hospitals, doctors, clinics don't have access to this, and it's not in the mainstream. This was the paper that we published about symptoms. Now, what is our experience of treating these patients? And I'll talk about how we've treated these patients a little bit. But really, we need to submit our treatment protocol to a randomized control trial, and then we can present the data to you. But essentially, we treated over 1,000 patients. We collected this information over 10 years. And like I said, it takes about 6 years for a patient to get to us. And if you look at their symptom profile, usually they will report, oh, I had a UTI, I got 3 days of antibiotics, it didn't work, I went back to the GP, I got some more, I then went back 2 weeks later, and so it goes on. They've had many investigations by the time they come to us, so cystoscopies, CTs, ultrasounds, all sorts of things. And various treatments as well, so urethral dilatations, distensions of the bladder, bladder installations. Now, 80% of these patients had pyuria. It's very interesting. Only 16% had a positive culture. Maybe we can understand a little bit why that might be now. And we collected data on their symptoms at each visit. They were about 54 years old. and they completed about 6-7 visits with us. We treated these patients with first-generation oral antibiotics, but at full dose. Now, how has this come about? This is, to be honest with you, through trial and error. We started by treating patients for 5 days, for 7 days, for a bit longer, then a bit longer, stopping, starting. And that's how we've actually essentially collected our data, including giving patients IV antibiotics, which I'll talk about at the end. The first generation aspect is important, and we're beginning to understand, now that we understand more about the microbiome, why that's important. So if you use antibiotics that are broad spectrum, you kind of tend to kill everything. And we now know that the bladder is not sterile, and that actually killing everything is probably not the right way forward. But really, there's some need to restore some sort of balance inside the bladder. We know a lot about how bacteria communicate with each other, largely from oral pathologists actually, and oral microbiologists, because they deal with this environment in the mouth. And really, it's not about killing something. When you kill everything, usually something horrible has the opportunity to come and take over. But I'm waiting for you all to gasp, but the average length of treatment for our patients was 383 days. Some less, some more, and depending on relapse. And we've sort of come to a protocol, and again, it's through trial and error. It was through stopping and starting and trying to get patients off. But this is a sort of a typical patient that we see, and you can see their LUTS symptoms. We use a validated 39-point score, and the LUTS symptoms you can see coming down nicely, and the white cells are reflected, so they sort of follow the same pattern. Now, IV antibiotics, I've put this in as the last slide because it's important, because a lot of our patients were going to accident and emergency, were admitted having IV antibiotics, and we tried IV antibiotics because we thought, is there a better penetration into the bladder with IV antibiotics? But what we found was really that you got better for a while, your symptoms started flaring again, and it's this sort of yo-yo effect. And we think this is due to the persisters, the persisting bacteria. So we've just stopped using IV antibiotics now, and our first-line regime is so much superior. So my message to all of you today, if nothing else, is to think about infection, understand the limits of our tests. More and more you will see it, our medical students will say, I would do a CT scan for every single thing that crops up in A&E. So again, UTI is the same. Dipstick, urine culture, microscopy, whatever, we have to be slightly suspicious. Listen to the patient, listen to their symptoms, they'll tell you. And be wary that you could be missing infection. Thank you.